Welcome back to Studious. In our last session, we mapped the branches and applied fields of microbiology. Today, we take a step back and ask a more fundamental question. How did we get here? How did humanity progress from having absolutely no concept of invisible life to building the most powerful molecular and genomic technologies in scientific history, all within roughly 3.5 centuries? The answer unfolds across 4 distinct eras. The discovery era of the 17th century, when the first primitive lenses cracked open the invisible world. The transition period, when scientists battled a 2,000-year-old philosophical myth about the origin of life. The Golden Age, when the rules of infection, sterilization, and immunity were systematically established. And finally, the molecular biology era, when microbiology descended from the organism to the gene and rewrote our understanding of life itself. This journey is not just history, it is the logical architecture of every concept you will study in modern microbiology. Understanding how each discovery was built on the one before it will make you a more coherent and confident scientist.
Before we explore the individuals and experiments in detail, let us establish the 4-era framework that structures the entire history of microbiology. Era 1 is the discovery era, characterized by the first direct observations of the unseen microscopic world, made possible by rudimentary but revolutionary optical instruments. Era 2 is the transition period, a philosophically turbulent time when the ancient and deeply entrenched doctrine of spontaneous generation was challenged, debated, and ultimately demolished by careful experimental evidence. Era 3 is the golden age of microbiology, a breathtakingly productive period of the 19th century, during which pathogens were cultured and identified, sterilization techniques were invented, the germ theory of disease was rigorously formalized, vaccines were developed, and antimicrobial compounds were first discovered. Era 4 is the molecular biology era of the 20th century, when the focus of microbiological inquiry shifted inward from the whole organism to its genetic material, leading to the elucidation of DNA structure, the one gene, one enzyme hypothesis, transduction, and ultimately the recombination DNA technologies that underpin modern biotechnology and genomic medicine. Each era did not merely add information, it fundamentally transformed the questions that scientists were asking.
The 17th century witnessed 2 observations that permanently altered humanity's understanding of life. The first was made in 1665 by Robert Hooke, an English natural philosopher and polymath who was one of the most gifted experimentalists of the scientific revolution. Using a compound microscope he helped design, Hooke examined a thin slice of oak tree cork and documented a remarkable pattern of box-like compartments, which he named celluli, from the Latin for small rooms, thereby founding the entire field of cell biology. His observations were published in his landmark illustrated work, Micrographia, which became one of the first scientific bestsellers in history. The second, and more microbiologically pivotal contribution, came from Antony van Leeuwenhoek, a Dutch draper from Delft with no formal scientific training, who through obsessive personal craftsmanship ground single-lens microscopes capable of magnifications up to 270 times, far beyond anything available to his contemporaries. Between 1674 and 1683, examining pond water, dental scrapings, and various infusions, Leeuwenhoek described living, motile microorganisms, bacteria, protozoa, and algae, which he called animalcules. His meticulous letters to the Royal Society of London represent the first authentic scientific record of living microorganisms, earning him his permanent title, the Father of Microbiology.
Following Leeuwenhoek's discoveries, a profound philosophical question arose. Where did these animalcules come from? The prevailing answer, inherited from Aristotle and unchallenged for nearly 2,000 years, was spontaneous generation, the belief that living organisms could arise de novo from non-living matter. Maggots from rotting meat, mice from grain stores, and now animalcules from infusions, all were considered self-generating. The first serious experimental challenge to this doctrine came from Francesco Redi, an Italian physician and poet, who in 1668 performed an elegant experiment demonstrating that maggots did not arise spontaneously in meat, but only when flies were allowed to access it and lay eggs. When meat was sealed in jars covered with gauze, allowing air contact but excluding flies, no maggots appeared. Redi's work was a foundational victory for the principle of biogenesis, the idea that life arises only from pre-existing life. However, Redi's experiments did not address microscopic organisms, and the spontaneous generation debate for microbes persisted for another 2 centuries. It fell to Louis Pasteur in 1859 and 1861, using his iconic swan neck flask experiments to definitively prove that microorganisms present in broth originated from airborne particles, not from spontaneous chemical generation, a conclusion that was the philosophical prerequisite for the birth of the germ theory of disease.
Even before the mechanisms of microbial growth were fully understood, practical innovators were developing empirical techniques to control and exploit microorganisms. Nicolas Appert, a French confectioner and chef, made a pivotal practical discovery around 1809 that would revolutionize food preservation and unknowingly lay groundwork for microbiology. Working in response to a challenge by Napoleon Bonaparte to develop reliable food preservation methods for the French military, Appert demonstrated that soups, vegetables, and meats sealed hermetically in glass jars and subjected to sustained boiling heat remained free from spoilage for months, a process we recognize today as the foundational principle of canning. Appert did not know that heat killed microorganisms. The germ theory had not yet been articulated. Nevertheless, his systematic observations that heat plus airtight sealing equals preservation was empirically correct and of enormous practical importance. Separately, German scientists Georg Schroeder and Theodor von Deutsch made a different but equally significant contribution. In 1854, they introduced the cotton plug into microbial culture tubes. This deceptively simple device, a dense wad of cotton sealing the mouth of a culture flask, allowed air to pass through while physically filtering out airborne microorganisms, enabling the first controlled sterile cultures of microorganisms in liquid media and directly facilitating the laboratory work of Pasteur and Koch that followed.
The second half of the 19th century represents the most intellectually explosive period in the history of microbiology, a golden age in which 2 towering figures, working largely independently, constructed the entire conceptual and technical framework of the modern discipline. Louis Pasteur, the French chemist and microbiologist, delivered the decisive blow to spontaneous generation through his 1859 swan neck flask experiments, in which sterilized nutrient broth remained clear indefinitely unless the curved glass neck was broken, demonstrating that contamination came from external airborne particles, not from the broth itself. Pasteur went on to develop the germ theory of fermentation, proving that specific microorganisms drove specific chemical transformations. He created attenuated vaccines against chicken cholera, anthrax, and rabies, and developed pasteurization, the application of controlled mild heat to kill pathogenic bacteria in liquids without destroying flavor. Working in parallel, the German physician Robert Koch pioneered the technical revolution in bacteriology, developing the techniques of solid culture media, pure culture isolation by the streak plate method, and bacterial staining. Koch identified the specific causative agents of anthrax in 1876 and tuberculosis in 1882, and formalized the rules of microbe disease causation in his landmark postulates. Together, Pasteur and Koch built the 2 pillars upon which all of medical microbiology stands.
The Golden Age was not only an era of discovery, it was also an era of decisive innovation in the control of microbial contamination, driven by the urgent practical demands of medicine, food science, and laboratory research. The first and most widely known sterilization innovation is pasteurization, developed by Louis Pasteur, which applies targeted mild heat, typically between 60 and 85 degrees Celsius, to liquids for a defined period, killing pathogenic vegetative bacteria while preserving the taste and nutritional quality of the product. Pasteurization transformed the safety of milk, wine, and beer and remains in universal use today. The second technique is Tyndallization, devised by Irish physicist John Tyndall in the 1870s, which addressed a critical limitation of simple boiling: heat-resistant bacterial endospores. Tyndallization uses intermittent fractional heating, boiling the liquid on 3 consecutive days with rest intervals in between. During the rest periods, dormant spores germinate into vegetative cells, which are then killed by the next round of boiling. This cycle effectively sterilizes heat-resistant preparations that single boiling cannot eliminate. The third innovation is antiseptic surgery, pioneered by the Scottish surgeon Joseph Lister, who in 1867 introduced the use of carbolic acid, phenol, both as a spray in operating theaters and as a wound dressing treatment. Lister's antiseptic technique dramatically reduced post-surgical infection and death rates, establishing the foundational principle of surgical asepsis that underpins every modern operating theater.
Behind the famous discoveries of the Golden Age lay a parallel and equally critical story of laboratory innovation, the iterative improvement of the physical tools and media used to grow and study microorganisms. Robert Koch recognized early in his work that liquid broth cultures were fundamentally inadequate for isolating pure bacterial species, because multiple organisms could grow together in suspension without producing distinguishable individual colonies. Koch's first attempt at a solidifying agent was gelatin, which had an obvious but fatal thermal limitation. It melted at temperatures above 28 degrees Celsius, far below the optimal growth temperature of most human pathogens, and could be liquefied by many bacteria themselves through gelatinase enzyme activity. The critical solution came from an unexpected source: Angelina Fanny Hess, the wife and laboratory assistant of Koch's colleague Walter Hess, who suggested the use of agar, a polysaccharide extract from seaweed that she used in her home kitchen as a setting agent for jellies. Agar melts only at approximately 95 degrees Celsius, but solidifies at around 42 degrees, making it thermally stable at incubation temperatures. It is also resistant to microbial degradation by most bacteria, and remains the universal culture medium solidifying agent in microbiology laboratories worldwide today. The final piece of the puzzle was provided in 1887 by Koch's assistant Richard Julius Petrie, who designed the shallow, lidded, circular dish that now bears his name, the Petri dish, providing the ideal contamination-controlled vessel for solid agar culture and colony isolation.
Robert Koch's most enduring intellectual contribution to science is not a single discovery, but a logical framework, a rigorous set of criteria that must be satisfied before any microorganism can be definitively accepted as the causative agent of a specific disease. Known as Koch's postulates, this 4-step logical chain was first articulated around 1884 and remains the foundational logic of infectious disease microbiology. Postulate 1. The suspected pathogen must be present in every case of the disease and absent from healthy individuals. Postulate 2. The pathogen must be isolated from the diseased host and successfully grown in pure culture outside the body. Postulate 3. The pure cultured pathogen, when inoculated into a healthy susceptible host, must reproduce the original disease in its entirety. Postulate 4. The pathogen must then be re-isolated from the experimentally infected host and shown to be identical to the original organism. Using this rigorous 4-step cycle, Koch identified the anthrax bacillus, Bacillus anthracis, as the causative agent of anthrax, the first definitive proof of bacterial disease causation, and then, in one of the most celebrated moments in the history of medicine, announced the discovery of Mycobacterium tuberculosis as the cause of tuberculosis on March 24, 1882, a date now commemorated annually as World Tuberculosis Day.
The closing decades of the 19th century and the opening of the 20th brought a cascade of transformative discoveries in immunity and chemotherapy that collectively constituted the birth of modern preventative and therapeutic medicine. Edward Jenner, working as early as 1796 in rural Gloucestershire, England, made the foundational observation that milkmaids who had contracted cowpox appeared to be protected against the far more deadly smallpox. Jenner's systematic experiments demonstrated that inoculating individuals with material from cowpox lesions, varioli vaccini, conferred protective immunity against smallpox. His word for this procedure, vaccination, is permanently rooted in the Latin vacca, meaning cow. Jenner's work predated the germ theory and any understanding of immune mechanisms, yet it established the empirical principle of vaccination that Pasteur would later extend to multiple infectious diseases. In parallel, Paul Ehrlich, a German physician and Nobel laureate, transformed the concept of disease treatment through his systematic search for what he called a magic bullet, a chemical compound that would selectively destroy a pathogen without harming the host. Ehrlich discovered trypan red for African sleeping sickness, and in 1909, salvarsan, compound 606, an arsenic-based compound effective against syphilis, launching the modern era of chemotherapy. The capstone discovery of this era arrived when Alexander Fleming, in 1928, observed that a contaminating Penicillin mold was producing a substance that killed bacterial colonies on his culture plate, penicillin, the world's first natural antibiotic and arguably the most important medical discovery of the 20th century.
The 20th century brought a seismic paradigm shift in microbiology. Prior to this era, microbiologists studied organisms at the scale of the whole cell, culturing them, observing their colony morphology, measuring their biochemical activities, and mapping their disease-causing behaviors. The molecular biology era fundamentally redirected this lens of inquiry, from the organism to the molecule, from the visible cellular body to the invisible genetic code inscribed within it. This transformation was catalyzed by a convergence of technological and conceptual breakthroughs. The development of ultracentrifugation, X-ray crystallography, radioactive isotope labeling, and eventually DNA sequencing. For the first time, scientists could interrogate the internal molecular machinery of microbial cells with extraordinary precision, mapping nucleotide sequences, elucidating gene regulation pathways, characterizing enzymatic reaction networks, and tracing the molecular signate transduction cascades that govern how bacteria sense and respond to their environment. The foundational philosophical shift was captured elegantly in the central dogma of molecular biology, articulated by Francis Crick in 1958, that genetic information flows from DNA to RNA to protein, a unidirectional flow of biological information that applies equally to bacteria, viruses, and human cells, and remains the organizing principle of all molecular life science today.
The molecular era of microbiology was driven forward by a remarkable generation of scientists whose experiments revealed the fundamental logic of genetic inheritance and gene function. Among the most conceptually powerful contributions was the one gene, one enzyme hypothesis, developed by George Wells Beadle and Edward Laurie Tatum through their elegant experiments with the bread mold Neurospora crassa in the early 1940s. By exposing Neurospora to X-ray radiation to induce mutations, Beadle and Tatum demonstrated that each mutation that disrupted a specific biochemical pathway could be traced to the loss of function of a single gene, and that each such gene corresponded to the production of a single specific enzyme. This work established for the first time the direct functional link between a gene and a biochemical product, earning Beadle and Tatum the 1958 Nobel Prize in Physiology or Medicine. Complementing this work, Max Delbruck and Salvador Luria, working with bacteriophages, viruses that infect bacteria, developed the mathematical and experimental framework for understanding microbial genetics. Their fluctuation test of 1943 proved that bacterial mutations arise spontaneously and randomly, independent of environmental pressure, establishing that bacteria possess true Darwinian genetics. They also revealed how core genetic material is faithfully and precisely transmitted from parent to daughter cells across generations, a discovery that earned them the 1969 Nobel Prize in Physiology or Medicine.
As the molecular era deepened, it became clear that bacterial genetics operated by rules fundamentally different from classical Mendelian inheritance. Bacteria do not reproduce sexually and do not undergo meiosis, yet they demonstrably exchange genetic information with extraordinary efficiency.
a phenomenon that confounded early geneticists. The resolution of this puzzle came primarily from the work of Joshua Lederberg, an American geneticist who made 2 Nobel Prize-winning contributions while still in his 20s. First, Lederberg introduced the concept of the plasmid to the scientific world, a circular, self-replicating DNA molecule distinct from and independent of the main bacterial chromosome, capable of carrying accessory genetic information, including antibiotic resistance genes, virulence factors, and metabolic enzymes. The existence of plasmids explained how bacteria could rapidly acquire new genetic traits not encoded in their chromosomal DNA and share them horizontally across even distantly related bacterial species. Second, Lederberg characterized the mechanism of transduction, a process by which bacteriophages act as unintentional genetic couriers, inadvertently packaging fragments of their bacterial host's chromosomal DNA during replication and injecting this DNA into a new bacterial host upon infection. Transduction represents one of 3 mechanisms of horizontal gene transfer in bacteria, alongside transformation and conjugation, and is a critical driver of antibiotic resistance spread in clinical settings, making Lederberg's work directly relevant to contemporary infectious disease medicine.
On April 25, 1953, James Watson and Francis Crick published a landmark 900-word paper in the journal Nature that would arguably become the most consequential single scientific publication of the 20th century, the double helix model of DNA. Drawing critically on the X-ray crystallographic data of Rosalind Franklin and Maurice Wilkins at King's College London, particularly Franklin's famous photograph 51, which revealed the helical B form of DNA, Watson and Crick proposed a structural model in which 2 anti-parallel polynucleotide chains are wound around a common axis, held together by specific base pairing, adenine with thymine and guanine with cytosine. This precise base complementarity was not merely an aesthetic feature of the structure. It immediately suggested the mechanism of DNA replication, whereby each strand could serve as a template for synthesis of an identical daughter strand, explaining for the first time the physical chemistry of hereditary transmission. The double helix model also established the structural framework for understanding gene expression, how the sequence of bases in DNA encodes the sequence of amino acids in proteins through the intermediary of messenger RNA. Watson, Crick, and Wilkins received the Nobel Prize in Physiology or Medicine in 1962. The model transformed microbiology, genetics, medicine, and forensic science, and directly enabled the recombinant DNA revolution of the 1970s and the genomic revolution of the 21st century.
This final slide captures the entire history we have explored today in a single ascending chain of discovery, and understanding this chain is the deepest lesson of this session. The ascent begins with seeing. Hooke and Leeuwenhoek revealed that the invisible world existed. Without that first revelation, nothing that followed would have been possible. The chain ascends to culturing. Appert, Schroeder, Koch, Hesse, and Petrie built the tools to isolate, grow, and observe individual organisms in controlled conditions. Without pure culture methodology, pathogens could not be identified. The chain ascends further to conquering. Jenner, Pasteur, Koch, Ehrlich, Lister, and Fleming used what they had cultured to develop vaccines, antiseptics, and antibiotics that transformed infection from a death sentence to a treatable condition. And finally, the chain ascends to programming. Watson, Crick, Beadle, Tatum, Luria, Delbruck, and Lederberg descended into the molecule and mapped the source code of microbial life, unlocking recombinant DNA technology, genomic sequencing, and the molecular medicine of the 21st century. 4 eras, each built perfectly on the shoulders of the previous one. Every breakthrough you will encounter in modern microbiology, every diagnostic test, every antibiotic, every vaccine, every gene therapy traces its ancestry back through this chain. If you found this video valuable, please like it, share it with a classmate, and subscribe to Studious. We will see you in the next one.